What if we could watch the exact moment a normal cell turns into cancer, like frame by frame, inside the human body?
Yeah, not a computer simulation and not a best guess.
Right, the actual biological tape playing out in real time. Because the messy, complicated transition from healthy tissue to a malignant tumor is usually, well, it's a black box.
It really is. You see the healthy tissue, and then, I mean, months or years later, you just see the tumor.
Exactly. But today, we are prying that black box wide open. So welcome to today's deep dive.
It's honestly a phenomenal leap in how we understand disease. The source material we are diving into today completely maps out what researchers call the precancer-to-cancer transition.
The PCT.
Right, PCT. And they're looking specifically at a disease called cervical squamous cell carcinoma, which we'll just refer to as CSCC for short.
Okay, let's unpack this, because before we get into the wild biological mechanisms they uncovered, we really need to set the stage. What makes this specific study so rare?
Well, historically, if scientists wanted to study how this cervical cancer develops, they had to compare healthy tissue from one patient with cancerous tissue from, a completely different patient.
Which seems like it would introduce a massive amount of genetic background noise. I mean, my healthy cells are fundamentally different from your healthy cells.
Precisely the problem. You get all these variables. But in this breakthrough study, researchers tracked the disease progression chronologically. So from normal tissue to high-grade lesions to carcinoma in situ, all the way to invasive cancer.
Wow.
And they did this within the exact same patients.
Just to pause on that medical term for a second so we don't lose anyone listening. Carcinoma in situ literally translates to cancer in its original place, right?
That's right, yeah.
Meaning the malignant cells have formed, but they haven't broken through into the deeper tissues yet. It's like they're sitting on the fence.
That's spot on. It is the crucial tipping point. So it's a true longitudinal look at how a tumor builds its home. Now, to understand CSCC, we have to recognize that it accounts for about 80% of all cervical cancers.
Okay.
And the primary driver for this disease is the human papillomavirus, or HPV, specifically high-risk strains like HPV-16 or HPV-58.
Those are the strains that are notoriously stubborn, right? The ones capable of integrating their DNA into our own cells.
They are. But here is the critical detail that drives our entire deep dive today. HPV infection alone is not enough to cause cancer.
Wait, really? I think a lot of people assume HPV just equals cancer.
Right, but the virus just initiates the spark. For a full-blown tumor to develop and thrive, the entire cellular ecosystem of the cervix has to fundamentally change. The surrounding tissue has to basically become a welcoming environment for those rogue cells to multiply.
So the neighborhood itself has to go bad, not just a single house.
Exactly.
As a listener, you might be wondering, how do you even begin to study a microscopic, tangled mess of millions of cells to see who is doing what? I mean, how do you map that crime scene?
That is where the methodology gets incredibly cool. The researchers relied on two massive cutting-edge data tools. The first is single-cell RNA sequencing, which we call cRNA-seq, and the second is spatial transcriptomics.
I want to make sure I'm visualizing this correctly. We know DNA is the master blueprint of a cell, but RNA is like the active instruction manual being read at any given moment, right?
Exactly. DNA is the library. RNA is the book you have open on your desk. It tells us what the cell is actually doing right now.
Okay, got it.
So single-cell RNA sequencing allows us to take a piece of tissue, break it down into tens of thousands of individual cells, and read that active instruction manual for every single one of them. In this study, they isolated over 31,000 high-quality cells.
I always think of it like this. Imagine you were trying to figure out the exact recipe of a really complex smoothie. Single-cell RNA sequencing is like having a machine that can identify every single molecule of strawberry, every speck of spinach, and every drop of almond milk. You get the ultimate definitive ingredient list.
I love that analogy. You know all the ingredients, but you have a blind spot, right? Knowing the ingredients in a blended smoothie doesn't tell you how they were arranged on the counter before they went into the blender.
You lose the architecture.
Exactly. You lose the physical architecture. And in a developing tumor, location is everything. Which cell is sitting next to which other cell? Who are they talking to?
And that is where the second tool, spatial transcriptomics, comes in. To stick with the metaphor, spatial transcriptomics is like looking at the unblended fruit bowl. You can see the whole bowl, and you know exactly which strawberry is resting against which spinach leaf.
Yes. By combining these two technologies, the researchers could see both the detailed genetic profile of every single cell and exactly where those specific cells were living in the tissue structure.
That is amazing.
It really is. And when they compared the unblended fruit bowls of the healthy tissue versus the cancerous tissue, they found a massive shift in the neighborhood population.
Let's talk about that big shift. What did the cellular neighborhood look like before, and what did it turn into?
In the normal cervix and in the early precancerous stages, the tissue is heavily dominated by two types of cells: epithelial cells, which form the protective outer lining, and fibroblasts.
And fibroblasts act as the biological scaffolding, right? The structural support.
Yeah, exactly. But as the disease progresses toward invasive cancer, something dramatic happens. The sheer number of fibroblasts and endothelial cells, which are the cells that line your blood vessels, starts to drop significantly.
Okay, so the scaffolding is breaking down and the blood vessel linings are decreasing. What is moving into the neighborhood to replace them?
Immune cells. An absolute flood of them. Yeah, we're talking T cells, B cells, natural killer cells, and monocytes, which are the white blood cells that act as your body's first responders to inflammation. In the early stages, these immune cells make up around 30% of the local cell population.
Okay.
But by the time it becomes full-blown cancer, they jump to over 60% of the entire tissue mass.
Wait, let me push back on that for a second, because if you are listening to this, you are probably looking at this flood of immune cells and thinking, shouldn't the body be winning this fight?
It's the logical conclusion, yeah.
Right. If your immune system, your natural defense army, is swarming the tumor area and doubling its presence, why on earth is the cancer still progressing?
It is the ultimate paradox of tumor biology. And the answer is that physical presence does not equal performance. Just because the immune cells are at the scene doesn't mean they are capable of doing their jobs.
So they're just, what, standing around?
Basically. What's fascinating here is that the tumor has actively reshaped its microenvironment to be heavily immunosuppressed.
So the immune cells show up to the battlefield, but their weapons have been confiscated.
Or their radios are jammed. The researchers discovered that it's actually the surrounding cells, the good structural cells that went bad, that are doing the jamming.
The fibroblasts and endothelial cells.
Exactly. As the cancer develops, specific subpopulations of those fibroblasts and endothelial cells change their identity. They identified a massive rise in what they call immune response cancer-associated fibroblasts, or IRCCAFs, and immune response endothelial cells, or IRECs.
Okay, here's where it gets really interesting. Because when we were going through the source material, a specific gene kept popping up in these corrupted fibroblasts and endothelial cells. The gene is called GNLY, which codes for a protein called granulosin. And I want to make sure we break this down clearly.
Granulosin is a completely fascinating player in this story. In normal human biology, granulosin is a weapon used by your immune system to destroy pathogens.
Right.
It literally punches physical holes in the outer membranes of bacteria and parasites. It is designed to be a killer protein.
Right. So logically, you would think, great, the tissue has lots of this granulosin protein, and it's going to punch holes in the cancer cells. But the data shows the exact opposite.
It does.
The expression of granulosin in these specific fibroblasts and endothelial cells strongly correlates with the tumor progressing faster. Why? How does a bacteria-killing weapon help a tumor?
This is where we have to look at the mechanism. When granulosin is appropriately used by an immune cell against a bacteria, it's a highly targeted strike.
Like a sniper.
Yes. But when these corrupted rogue fibroblasts constantly pump granulosin out into the tumor microenvironment, they aren't aiming at a specific bacteria. They are just flooding the entire zone with a highly toxic inflammatory chemical.
Ah, so it's friendly fire.
Exactly. Yeah. This chronic, inappropriate exposure to granulosin essentially chemically exhausts the T cells that arrive to fight the cancer. The toxic environment triggers apoptosis, or programmed cell death, in the very immune cells trying to help.
Wow.
It's like the body called in the fire department, but the firefighters are pouring gasoline on the fire.
That paints such a vivid and, frankly, terrifying picture. But this immune failure is happening on multiple fronts, isn't it? It's not just the T cells getting burned out by granulosin. The B cells are failing too.
Yeah, they are.
And B cells are the ones that act as our antibody factories, right?
Correct. The researchers tracked the developmental stages of B cells in the tissue and found a catastrophic drop in a subset known as progenitor B cells.
Progenitor meaning like the early ones.
Right. Think of progenitor cells as the trainees. They are early stage cells that are supposed to grow up, mature, and differentiate into fully functional antibody factories. And these trainees are characterized by the expression of a specific gene called IGFBP7.
And as the precancer transitions into invasive cancer, the expression of that IGF-DP7 gene just plummets.
It drops off a cliff. The body is physically failing to manufacture mature B cells properly. The biological assembly line is basically broken.
Okay, let's just take a breath and recap what we've covered so far, because this is a lot of dense biology. So imagine you're looking at the cervical tissue. You have a massive flood of immune cells arriving at the cervix, right? But the T cells are being chemically exhausted by rogue fibroblasts pumping out granulosin.
Yep, the friendly fire.
And at the same time, the B cell assembly line is completely shut down because they lose that IGF-BP7 gene. So the local immune response is entirely paralyzed.
It is a masterclass in cellular manipulation. The tumor doesn't just fight the immune system, it turns the neighborhood against it.
That is just wild. But knowing that these cellular players are corrupted is really only half the battle. If you are a clinician listening right now or a patient, you're probably wondering, how do we actually use this information? Is there a specific molecular fingerprint doctors can look for to know if a patient's precancer is about to turn into full-blown cancer?
There is. And identifying that fingerprint is one of the most vital clinical outcomes of this deep dive. Out of all the tens of thousands of genes they tracked, using that single-cell RNA sequencing, 3 critical suspects emerged as the ultimate predictors of cancer.
A biomarker trio.
Exactly. CLDN1, ISG15, and PTGDS.
Let's break down this trio. Who are they and what are they doing in the tissue?
Let's start with PTGDS. Think of this gene as the lost good guy. In a healthy cervix, PTGDS acts as a tumor suppressor.
Okay.
A tumor suppressor is basically the brakes on a car. It tells cells to stop dividing before they get out of control. But the researchers found that as the precancer transitions to invasive cancer, the expression of PTGDS drops dramatically.
The brakes just disappear from the vehicle.
Exactly. And what's remarkable from a diagnostic standpoint is that they found high levels of PTGDS in a patient's blood serum could actually mean a better prognosis. In their testing, it had a massive 92.31% positive detection rate in patients.
So its sudden absence is a glaring, massive warning sign that the cancer is accelerating.
Exactly. It's a huge red flag.
So that is the lost good guy, the brakes. What about the other two, CLDN1 and ISG15?
They are the bad guys. In genetic terms, they act as oncogenes. If tumor suppressors are the brakes, oncogenes are a stuck gas pedal. Yeah, they actively promote cancer growth. And unlike PTGDS, which disappears, the expression of CLDN1 and ISG15 absolutely spikes during the transition to invasive cancer.
Let's ground this in reality for a second. Imagine you are a patient or a doctor dealing with a routine cervical biopsy that shows high-grade precancerous lesions. Right now, it can be really difficult to predict whose lesion will stay dormant for years and whose will rapidly progress to aggressive cancer.
And one of the biggest challenges in oncology.
Right. But by combining the detection of these 3 specific genes, looking for the drop in PTGDS and the spike in CLDN1 and ISG15, you could theoretically identify high-risk patients immediately. You could intervene early before the cancer ever truly breaks through the basement membrane.
It shifts the entire medical paradigm from reactive treatment to proactive interception.
Which is incredible.
But of course, identifying the genes is great for diagnosis. The next logical step, and what the researchers spend a massive amount of time on, is understanding the mechanism.
Yes, because knowing they're at the scene of the crime is one thing, but how exactly are these genes committing the crime? Like, how does a spike in ISG15 actually cause a tumor to multiply?
To explain this, we have to look very closely at the protein ISG15. This is where the spatial transcriptomics data, the unblended fruit bowl we talked about, becomes absolutely crucial.
Right, because it shows location.
Exactly. Because it showed that ISG15 isn't just floating around randomly in the tissue. It is specifically being secreted by those corrupted inflammatory fibroblasts we talked about earlier.
Wait, the same ones that were exhausting the immune system with granulosin?
The very same ones.
Wow, they are busy.
They are very busy. So these inflammatory fibroblasts secrete the protein ISG15 into the space immediately surrounding the tumor. Now, ISG15 is what biologists call a ubiquitin-like protein. And in normal cellular biology, ubiquitin is famous for being the kiss of death.
The kiss of death. How does that work?
When a protein in your cell gets damaged or is no longer needed, it gets chemically tagged with ubiquitin. That tag is a signal for the cell to transport that protein into the cellular trash can, a structure called the proteasome, where it gets shredded and destroyed.
Okay, so if ISG15 is like ubiquitin, is it destroying things around the tumor?
What's fascinating here is that it does the exact opposite. ISG15 binds to a specific growth factor in the tissue called FGF1. But instead of tagging FGF1 for the cellular trash can, it stabilizes it. It fundamentally alters the structure just enough to protect it from degradation.
So ISG15 is essentially acting as a cellular bodyguard.
That is a perfect way to visualize.
It throws itself in front of the cellular trash compactors to save the FGF1 protein.
It acts exactly like a shield. And because FGF1 is protected by its bodyguard, it survives in the treacherous microenvironment long enough to travel over to the actual cancer cells.
Okay.
Once there, it binds to a specific receptor on the surface of the tumor cell, which is called FGFR1.
And what happens when FGF1 docks into that FGFR1 receptor on the tumor?
It acts exactly like a key turning in an ignition. It activates one of the most notorious, well-studied cancer growth signaling cascades in all of human biology, the PI3K-AKT-mTOR pathway.
Okay, that is a serious alphabet soup right there.
Yeah.
Let's translate that pathway into plain English for the listener. What is actually happening in the cell when this pathway turns on?
Sure. Think of it as a cellular chain of command. PI3K is the radar on the cell membrane. It detects the growth signal from the outside.
The radar, got it.
Once activated, it passes the message to AKT, which is the messenger sprinting through the cell to the control room.
Okay.
Finally, AKT activates mTOR, which is essentially the factory floor. mTOR physically drives the building of new proteins and materials.
So it's radar to messenger to factory floor.
Exactly. When this pathway turns on, it forces the cancer cell to survive, to metabolize energy rapidly, and to aggressively multiply.
So putting it all together for you guys listening, the corrupted neighborhood fibroblasts secrete the bodyguard ISG15. That bodyguard saves the growth factor FGF1, allowing it to run over to the tumor cell, turn the ignition switch, and tell the factory floor to multiply.
You've got it completely. And this proves that the cancer isn't growing autonomously on its own.
Right.
There is direct physical crosstalk between the surrounding corrupted fibroblasts secreting the bodyguard and the actual tumor cells receiving the growth signal. The healthy tissue has literally been hijacked to actively feed the tumor.
That changes the way we have to look at the disease entirely. Because if we know the exact communication pathway the tumor uses, if we know exactly how the neighborhood is making the phone calls to the tumor, that actually makes me wonder, how do we cut the phone lines?
That exact question brings us to the therapeutics phase of the study. Once the researchers mapped out this bodyguard mechanism, they had to prove they could actually stop it.
Right.
To do this, they ran extensive tests, both in vitro, meaning in petri dishes, and in vivo, using living mouse models.
Okay, it's one thing to see this in isolated cells on a piece of plastic. To prove this, you'd have to replicate this exact cellular neighborhood in a living body. What happened in the mice?
Well, they genetically engineered the tumors in the mice. When they artificially forced the tumors to overexpress CLDN1 or ISG15, the tumors grew massive and rapidly. The biology behaved exactly as predicted.
And when they stopped it?
When they silenced those specific genes, when they genetically knocked them down, the tumors shrank significantly.
But obviously, we can't easily genetically engineer a human patient's tumor in the clinic yet. So are there actual existing pharmaceutical drugs that can target this specific pathway right now?
This raises an important question about translating research to the bedside. And the answer is yes. Because that PI3K-AKT-mTOR pathway is so infamous in cancer biology, pharmaceutical companies have already developed clinical inhibitors for it.
Oh, that's incredibly lucky.
It is. The researchers decided to test several of these existing drugs on these specific cervical cancer models to see if they could break the chain of command.
Let's name names. What exactly did they test?
And tested alpelisib, which is designed to inhibit PI3K, the radar. They tested capivasertib, which blocks AKT, the messenger. And they tested everolimus, which blocks mTOR, the factory floor.
And did they work?
All of them showed varying degrees of success in slowing the cancer down by jamming the signals.
But the data takes a turn here, right? There was one drug that completely outperformed the rest.
Yes. The real star of the show was a compound called PD-161570.
Catchy name.
Very catchy. This drug doesn't bother targeting the downstream signaling inside the cell. It goes right for the ignition switch on the outside. It is an FGFR1 inhibitor.
Oh, so it blocks the receptor itself.
Exactly. It physically blocks the exact receptor that the FGF1 growth factor tries to bind to. In the animal models, PD-161570 showed massive tumor suppressive effects. It fundamentally proved that if you block this specific pathway, you can cut the fuel line to the cancer.
It stops the key from turning in the ignition in the first place. That is incredibly promising. But what does this mean for patients right now, today, before these specific targeted therapies become the standard of care for cervical cancer? I mean, does knowing this genetic fingerprint help a doctor make decisions today?
It does, significantly.
Yeah.
Beyond just diagnosing the cancer, the researchers looked at tissue microarray data. These are basically large clinical databases of tissue samples linked to actual patient outcomes over years.
Okay.
They found a sobering but vital reality. Patients whose tumors had naturally high levels of ISG15, or CLDN1, had significantly reduced overall survival rates. Their cancers were mathematically more aggressive.
So it serves as a prognostic marker. It tells you exactly how dangerous the specific cancer in front of you is.
It goes even further than that. The exploratory data strongly suggests that high levels of these specific genes might predict a poorer response to immune checkpoint blockade therapy.
Which is a very common type of modern immunotherapy, right?
Extremely common, yes.
So what does this all mean for you as the listener? It means knowing this precise genetic profile doesn't just help diagnose the cancer. It dictates exactly how a doctor should and shouldn't treat the patient.
Absolutely.
If you know a patient has sky-high levels of the bodyguard protein, ISG15, you might know immediately that standard immunotherapy isn't going to cut it, and you need to pivot immediately to aggressive, targeted inhibitors instead.
If we connect this to the bigger picture, it is the absolute definition of personalized medicine. We are moving away from a blind, one-size-fits-all approach to oncology and moving toward treating the specific, unique cellular mechanics of the tumor in front of you.
That is just incredible. Let's take a quick breather and look at the entire landscape we've covered today, because we have traveled through a lot of biology. We started by mapping the changing landscape of the cervix using spatial transcriptomics, watching as the healthy tissue scaffolding was broken down and overrun by a massive flood of immune cells.
But those immune cells were paralyzed. We discovered the culprits were corrupted fibroblasts and endothelial cells expressing granulosin.
Right, which chemically exhausted the T cells and shut down the B cell assembly line entirely.
From there, we uncovered the 3-gene fingerprint, the dramatic drop in the good guy tumor suppressor, PTGDS, and the massive rise of the bad guy oncogene CLDN1 and ISG15.
We learned how ISG15 acts as a cellular bodyguard, protecting a growth factor from the cellular trash can so it can trigger the PI3K-AKT mTOR pathway and tell the cancer's factory floor to multiply.
And finally, we saw that by mapping this exact communication pathway, we can deploy targeted inhibitors, like FGFR1 pluggers, to cut the communication lines and physically shrink the tumors, all while using the genetic profile to predict survival and guide immediate treatment choices.
It is a phenomenal piece of biological detective work, and it leaves me with a final provocative thought that I want to pass on to you, the listener, to mull over. For decades, traditional medicine has thought about treating cancer by attacking the tumor cells directly.
Right, the search and destroy method.
Exactly. We try to poison them with chemo, burn them with radiation, or physically cut them out with surgery. But this deep dive shows us that the tumor is never a lone wolf. The surrounding healthy tissue, the fibroblasts, the blood vessels, they are the ones pulling the strings.
Yeah, they're providing the bodyguards and jamming the immune system. They are the architects of the tumor's ecosystem.
Exactly. So here is the thought. If we can develop ISG15 inhibitors or drugs that artificially activate the tumor suppressor PTGDS, could we one day treat cancer without ever targeting the cancer cells themselves?
Wow.
Could we simply re-educate the healthy tissue around a precancerous lesion, shifting the ecosystem so drastically that the tumor never even has a chance to form in the first place?
It's a profound shift in perspective, moving from trying to destroy an entrenched enemy to simply making the terrain uninhabitable for them before they can even build a home.
It flips the whole script on oncology. Thank you so much for joining us on this deep dive. Keep questioning the world around you, keep looking for the hidden connections, and we will see you next time.